Okay, so then what happens is, guys, we are going to take this drop, we're going to put it out over here onto this medium. Now, basically, this medium, this is nothing but your solid medium. Okay, we said we are going to use two mediums. One is the NOPS medium, and one is your MS medium, that is your Murashige and Scooby medium. Okay, so either of the two mediums. Then what happens the moment we put a drop out over here, all those cells, they're going to get absorbed into this medium. So what happens? The medium is going to supply all the nutrients to those cells. As a result, those cells, they are going to start multiplying out over here. They start dividing and dividing, dividing and dividing, dividing and dividing. As a result, what are we going to get out over here? We are going to get a small undifferentiated mass of cells. That small undifferentiated mass of cells, guys, what do we call this thing? We call this thing as the callus. Okay, but now what happens is in this entire medium, guys, we are going to add your hormones, that is your auxins and your cytokinins. Okay, and always remember these auxins and cytokinins, they are going to have a ratio. Okay, and because of that, we are going to see either the shoot formation first or the root formation first. If we have shoot formation, then it is your collagenesis. If we have root formation, then we call this as your rhizogenesis. So depending upon what you want first and adjusting the concentrations of these hormones, okay, you can get whatever part that you want first. Okay. Now, when you allow this callus to grow, guys, what is this going to grow? This is going to grow into a seedling, or this is going to grow into a sapling. Now, understand, guys, this is only from this one drop of cells. Like this, we have this entire vessel full of cells. And from this entire thing, we can convert, like, hundreds and hundreds of plants from this one entire explant. So this is why this entire tissue culture technique is really important. Guys, there are two things. If we make use of this liquid culture, then it is called as suspension culture. And if we take the explant and we directly put it onto the solid medium, then this is known as a solid culture. However, generally we follow first, it is going to be put into your suspension culture, and from suspension culture, we are going to follow it this way. Okay. So afterwards, like, guys, we've talked about, like, what are the medias, what is the pH, how we are going to maintain it, so on and so forth. So we've talked everything about this. After that, we've talked about this laminar airflow. Like, what did we see? One of the most important requirements of this experiment is to maintain an environment which is going to be organism or microorganism free environment. Okay, and how that can be maintained? That can be maintained only with the help of your laminar airflow. So what did we say, guys? In this laminar airflow, we are going to get some filtered air, okay, which is going to be passed through your HEPA filters. So this is going to be pure air without any microorganism. And plus, before we start off with any kind of experiment, whatever instruments, equipment, whatever that you're going to require, what do? We are going to keep it out over here in that laminar airflow. We have a UV tube light out over there. So what do? We switch on the UV tube light at least for a period of, like, 15-20 minutes before we start with the experiment. What does this do? This is going to kill all the germs which are present in the air, in the laminar airflow, on your test tube, everything. Okay. So all that thing is going to happen out over here, guys. Okay, once 15-20 minutes have passed by, then what are you going to do? You're going to switch off this UV tube light, and then you are going to start working out over here. Remember, you are not going to keep your cell solution out over here. Okay, you are just going to keep all the equipments and the sterile media, whatever that is there, only that part. You will not keep the cells. If you keep the cells of the plants out over here and expose them to UV, then immediately you're going to get mutated cells. That is not what we are looking for. Okay, right? And then after that, like I said, the callus culture, that is a solid culture, and we have the suspension culture. Okay. And finally, guys, we talked about, like, some of the applications out over here. Like, why are we going to go for this thing? Like, why is micropropagation important? Because it is going to help in like, rapid production of your plants. Apart from that, what is it going to do? It is going to make the plants disease free. Why? Because we have taken it from your apical meristem, and as I told you, apical meristem is always going to be disease free. After that, we said that, we have all these secondary metabolites which are going to be produced. Guys, very, very important. These are going to be asked in your exam. Okay, So be a little careful of this. One of the most important thing is, guys, this is going to make the plant season free. Okay, why is it going to make the the plant season free? Because see, in your laboratory, you can maintain whatever conditions you want. Okay, the desired condition, and if you give all the proper conditions to the plant, okay, this seedling, sapling, whatever that is growing in the test tube, immediately the plant starts becoming slowly season independent. So just like how we have mangoes coming only and only in your summer season, you can still get mangoes throughout the entire year. Okay, however, but for that we still are a little far away. But then that is the idea behind it. Okay, if by any chance there is only one plant or any some rare endangered species and you want to save it, then this is one of the best techniques ever because with this you can generate like thousands and thousands of plants from even a small single tissue. So that is why this is very important technique for your rapid propagation of any rare and endangered species. And finally, we said that guys, we have this somatic hybridization. Okay, so basically, when we talk about your somatic hybridization, what did we say, guys? We take two cells. Okay, and we are going to take only the protoplast from those two cells. What is a protoplast, guys? When the cell wall is removed and only the cell membrane is remaining, that is called as the protoplast. You take these two cells, you going to just put them together, and then these two cells they are going to mix together. That kind of fusion, this is called as your protoplast fusion. But now what happens, guys, because of this fusion you are going to see that both the characteristics they are going to combine together directly. And then what did we get from this thing, guys? We talked about your potato and tomato. When we take them and we bring for this somatic hybridization, we are going to get your tomato. And then we have Raphanus and we have your brassica. We get Raphano brassica. Okay, so this is what we had talked about in the last class, guys. Okay, but now what I want is I want you to just quickly go through this thing, see whatever we talked about. Okay, whatever we have talked about, like sorry, how this thing is going to be, like what really is going to happen, how we are going to bring about this thing. All that thing can be summarized in one small simple chart. Okay, guys, remember this chart. I have taken this thing from your textbooks only. Okay, so it is nothing different. Whatever we have talked about, in all that entire process, just it is going to be written down in your a few this thing in your few points. Okay, chalo. So if you want, we'll just quickly browse through that also. What are the points? And then we have a very small part to be done. That is, once again, we talk about single cell proteins, and then we are done with your entire chapter. Okay, so guys, first of all, like I said, before we start with your experiment, we need, oh, 1 minute. Okay, what we need is we are going to need all your equipments. Okay, whether it is your glass pipettes, whether it is your test tubes, whether it is you're going to be your rack, whether it is going to be any kind of your tips, whatever it is, whether it is any flash or your media, whatever it is, we need to get it in the most purest form. That is, it should be autoclaved. Okay, autoclave meaning we're going to put it in an industrial cooker. So if you remember, I talked about that. So, okay, so we have taken all your utensils, we have taken your equipments, instruments, we put it in an autoclave. Okay, now what does this autoclave do? Autoclave is going to kill all the bacteria. Okay, whatever it may be, it is going to kill it. How is it going to kill it, guys? Because in this autoclave, we are going to keep all the medium, like everything, whatever that is there, for a period of like 20 minutes. Okay, for a period of 20 minutes in your 15-pound pressure. Okay, is remember these values. Okay, it is 15-pound pressure. Okay, for a period of 20 minutes and 15-pound pressure. However, in real life, we really don't follow these, values because, to keep this autoclave, okay, minimum you are going to require like, 40 to 50 minutes to run this entire thing. So generally 20 minutes, we just say, okay, it is 20 minutes. This is the standard operating procedure. But in reality, it doesn't work that way. Right? Same way, we are going to do the same thing with this MS medium also, Murashige and Scoop. So whatever you want, you take that medium, put it into your flask, and then you take those flask and put it into the autoclave. We are going to sterilize everything. Now, once this everything has been sterilized, this is ready for our experiment. Now we shift our focus onto the plant material. So we are going to take that explant, whatever that we have, and now we are going to start working with this explant. So we are going to remove it first. Removal, before removal, what are we going to do? We need to sterilize it. So what are we going to do, guys? We are going to take some alcohol, or we are going to take some hypochlorite, or we are going to take some mercury chloride, and we are going to sterilize the surface of the plant. Then we are going to cut it, and then we are going to see. So we are going to sterilize the plant, then we are going to remove it, and then we are going to take it. We are going to cut it or excise it from the plant. Okay. Now, see, after this, we are going to take this inoculation. Inoculation means what? Inoculation means addition, basically. So these days, you must be hearing inoculation of germs into your body. What is that? Vaccination. Okay, so inoculation is nothing but taking these cells from the plant and putting it into your medium, that is your culture plus. Okay, medium, culture plus medium. Okay, now as I said, you're going to keep it out over here. Now you're going to allow the medium to rotate by putting it on a shaker, and during which you're going to see that the cells, they are going to start growing. As a result, we take some of the cells from here, like, you take a drop of the solution, and then we are going to put it onto your this thing, solid medium. What is it going to give rise to? It is going to form callus. What is this callus? Undifferentiated mass of cells. So now we are going to take this callus, and now this step, it is, like, if the callus needs to be like, kept for a very long period. Okay, so in that case, something that we call this like subculturing. So what we'll do is we'll take some cells from there, we'll put it onto another medium, take some cells from there, put it onto another medium, and this has to be kept on. So as to maintain the viability of the callus. So guys, here also the same thing. The callus is going to be divided into 3-4 segments and transferred into fresh culture mediums. Immediately you are going to see that the medium is going to be supplemented with your oxygen and cytokinins, and that is going to help in your organogenesis. Organogenesis is nothing but organ formation, and depending upon that, guys, you're going to see your root formation and your shoot formation, rhizogenesis and your collagenesis, and ultimately this is going to give you your small plantlet, seedling, or sapling. Okay. Finally, guys, what are we going to do? These plantlets, they are going to grow into the small test tubes. From small test tubes, they're going to be transferred into polythene bags, okay, containing your soil. Okay, and then their growth is going to be monitored. Once they are ready, once they have like grown really properly, we are going to take them and we are going to transfer it to the field. So that way. So this is the entire process, okay, of how this entire experiment of tissue culture really works. Okay, everybody all right with this thing, guys? Okay, everybody okay with this? Chalo. Okay, so guys, let's try to move ahead and let's try to see once again the last part. Okay, the last part is nothing but your single cell protein. Now, actually, we have talked about single cell protein when we were talking about mushrooms and all, guys. We spoke of this thing. What are these single cell protein? Basically, these single cell proteins, they are going to be like, really, really rich in their protein content. So generally what we do, we are going to take them, we are going to dry them, and then we are going to eat them as a food supplement. So who are we really talking about? We are talking about your spirulina, we are talking about your chlorella. Okay, so we are talking about those, algae out over there basically. Okay, but now single cell proteins, as I said, they can be of different types. Okay, so we talked about, like, it can be your Bacillus subtilis also, it can be your Candida also, it can be your Cyanobacteria also. Okay, so basically it can be anything. But now let's try to understand the reason why did we go for this single cell protein and what really happened out over there. Just a second. Okay, so now what really happened is, see, guys, now as the population increases, okay, as population increases, what happens? The demand for food is always going to increase. But there are going to be certain cases where we are not going to be able to meet the demands properly because, see, what happens? The food is always going to grow in your arithmetic progression, and population is always going to grow in your geometric progression. Okay, so let me just, okay, so food and your population. Okay, so both of them, they are going to grow in two different ways. So food, it is arithmetic progression, and this is geometric progression. Okay, so what is the difference between the two? Okay, so basically, when we talk about this arithmetic progression, guys, how this food is going to increase in its amount, it is going to be 1, 2, 3, 4, 5, 6, 7, 8, and so on and so forth. It grows this way. But guys, when we talk about your geometric progression, initially it is going to be 1. Okay, so 1 is going to go into 2. Okay, first. Guys, here until here, this is going to remain the same. After this, everything is going to change because this is exponential. So 1, 2, 2 is going to go into 4, 4 is going to grow into 8, 8 into 16, 16 into 32, 32, 64, 64, 128, 256, and so on and so forth. So, guys, if you look out over here, there is always a discord between the amount at the rate at which the food is growing and the rate at which the population is growing. So, slowly, slowly, guys, it becomes really, really difficult to feed the population. And while it becomes difficult to feed the population, it also becomes difficult to maintain or take care of their daily requirements. And one such requirement is proteins, because proteins are going to be required in large quantities because they are going to be your building blocks of the body. Okay, fine. So now what happens? So guys, so traditionally, what has been happening now? See, mostly people who are going to be non-veg, where are you going to get your source of proteins? You're going to get it from your animals. Okay, so basically, whether it is your chicken, whether it is your mutton, whether it is going to be eggs, these are going to be really, really rich in your proteins. Okay, so we generally go for those things. But now, those who are vegetarians, how are they going to get their things? So you're going to have sprouted, beans, or it can be your sprouted moong, or sprouted dals and all. So basically, you are going to go for that. Okay, fine. Apart from that, we also look out for your dairy products. Like if you talk about your cheese, if you talk about your paneer, we are talking about your milk as such. So we are going to look out for that as well. But now, guys, let's say we are talking about your this thing. Okay, so let's say we are talking about your chicken. Okay, or let's say we are talking about your mutton. Okay, a goat. So what happens is, guys, in order to get 1 kg of meat, 1 kg of meat from your mutton, you actually need to invest a lot more of food and water into this animal so that we will get that 1 kg of meat. Because see, you have to continuously feed that animal. So you are utilizing your resources, plus you are giving water daily, day and night to this animal. So once again, you are stressing onto your water resources. So what happens is, guys, you to feed such a huge population, your resources are constantly under stress. And slowly, this entire sustenance is not going to be good enough, and we won't be able to tolerate it anymore. So now what happens is we need to look out for some other way by which we can still take care of the nutritional requirements of the masses. And that is where we come up with your single cell proteins. The reason for the single cell protein is because there is too much amount of investment in terms of your food, in terms of your water, in terms of your efforts in order to generate little amounts of your meat. That is why we will go for your SCP, single cell protein. Okay. Now, guys, once again, the single cell protein basically, okay, this is not a new field. It is like, a very old field only. However, in the past few years, we have seen like, tremendous increase in its promotion, in its, application, in its marketing, in its distribution and all. So then it has become like, really, like, proper. Okay, so basically here, guys, when I talk about this SCP, what does this single cell protein actually mean? Like, what is it exactly going to be? Now, guys, you can directly have cells. Okay, some of the cells they're directly edible, so you're going to dry them, you're going to kill those cells, and then you're going to eat them. So they're going to be directly a source of your proteins. So one, or what is this? This can also refer to it refers to the crude. Okay, it refers to your crude or edible protein, the crude or the edible protein which is extracted from the cells. Okay, that is your microbial cells. Okay. And- okay, extracted from microbial cells, or we can say your dead cells, basically dead or dried cell biomass. Okay, because see, once again, what do I mean by this, dried cell biomass? So, for example, when we talk about your spirulina, now, guys, generally, people will go for your spirulina tablets and all. It is really, really rich. It has various kinds of your vitamins, various kinds of your proteins inside. So, it's like really, really rich. But, guys, the drawback of these spirulina tablets, okay, the ones that I had tasted years and years ago, is its taste. Its taste is like, really, really horrible. It is horrific. Guys, if you have this thing once, the taste is going to linger onto your tongue for the next 2 days. And the taste is like that of a rotten chicken. Okay. It was a really, really deadly stuff. So I tried it, and after that, I've never even tried it ever again. So it is that way, guys. Okay, but then once again, if you have this spirulina, spirulina is going to grow in your waters basically your water resources. Okay, now what do you have to do? You are going to take those, spirulina, you're going to dry it because we cannot just have it that way. So you're going to dry it. The moment you dry it, you're going to kill it, and then that is going to be collected and converted into a tablet. That is how it works with your spirulina. Okay, so basically, that is why we say that that is nothing but your dried cell biomass. Okay. But now, as I said, who are the ones which we are going to use? We can use your algae, we can use your yeast, we can use bacteria, we can use fungi. All of them, they are going to be really, really rich in their protein content. Okay. And how we can do this thing? Once again, we can grow this thing on your, like, some microbiological media. And now, as they grow, like, sometimes we have like very simple solutions. Like, for example, we have we have a water body, and I want to grow spirulina out over there. So what do I do? I'm going to take some amount of your molasses. I take a mixture of some amount of like, food particles and all mixture, and then I'm going to add it into this water body. Okay, water body meaning I'm saying like, it's a small pond, or it is going to be a small like, a tank kind of a thing that has been made. Okay, and now you're going to just stir it and you're going to leave it as it is. You will see slowly your microbial growth taking place on it. Okay, so this is how we can do it, like, with a minimum amount of investment as well. Okay, but now, guys, what happens is these organisms, okay, they are like, really active, okay, and they are going to be really fast. So we have two important things. So, guys, already we have spoken of like a single cell protein. So I'm not putting down everything out over here again because already there it is in the short note of your mushrooms. Okay, but now, guys, who are the ones that can be used as your single cell proteins and what is the importance behind them? Right? So, guys, we said we are going to have fungi, we are going to have bacteria, we are going to have your algae. Okay, so let's try to see in your fungus who can be used as single cell protein. So, first of all, guys, don't forget we have your mushrooms. Somehow your textbook has forgotten about mushrooms. Okay, anyways, after your mushrooms, we can go for your aspergillus. Okay, so aspergillus niger. Guys, if you remember, we had your citric acid, gluconic acid, and the same aspergillus niger. Okay, along with this, we also have your trichoderma. Okay, but now this is not Trichoderma coligi, this is Trichoderma viride. Okay, the second name, even if you don't remember, that is still fine, but you need to remember the first name at least. Okay, after after your this thing, we are going to have your algae. Okay, like I said, we have algae. So which algae are generally used for SCP production? So as I said, we are going to have spirulina, and I talked about the other one also, that is your chlorella. So spirulina and chlorella. Okay. After this, we move on to your yeast. Sorry. Okay. So in your yeast, guys, I said we are going to use two. So one is Saccharomyces cerevisiae, okay, which is nothing but our normal yeast. Okay. So the baker's yeast that we call it, the baker's yeast, the brewer's yeast. Okay, that is your Saccharomyces cerevisiae. And the second one in the same category, guys, we are going to have Candida, Candida utilis. Okay, and the last one, guys, we are going to have our bacteria. And guys, this name is very important. Okay, Methylophyllus. Okay, we have Methylophilus, Methylotrophus. Guys, a huge name, but very important. Okay, Methylophilus, Methylotrophus. It's a single name. L- Last fungus is Trichoderma viride. Okay, and guys, the next one is we are going to have something that is your bacillus, but this bacillus is a little different. This is Bacillus megastabium. That way. Okay, so guys, these are the different varieties that we can have. Like I said, guys, these things they do not require much amount of your requirements to grow. Even if there is one small pond or one small lake, even there it can start growing. Okay, the out of this, guys, this Methylophilus Methylotrophus, guys, remember, this is like one of the most rapidly dividing single cell protein. Guys, in one day, okay, in one day, I'll just give you an idea. If you feed a cat, if you feed a cattle, basically, let's say you're talking about a cow, okay, so you feed a cow throughout the day, taking care of all its nutrients and all. At the end of the day, you may experience that, okay, maybe the cow's weight has increased by 1 kg. So that much amount of effort and work is going to go into generating 1 kg of your meat. Okay, let's imagine. But now, If we use Methylophilus methylotropis, guys, what you are going to see is that this thing by the end of the day, okay, at the start of the day, you have like a little quantity, you have just grown it into a typical media and let allow it to grow. By the end of the day, guys, this is capable of giving rise to 1 ton kg of your single cell proteins. So guys, this is like the most important, okay, most important bacteria ever, single cell bacteria. Okay, but guys, let's try to see a few advantages. Okay, why is this, so advantageous, or why is this very lucrative business in today's time? So let's try to see. Okay, so guys, what did I just tell you about Methylotropus? Okay, Methylophilus methylotropus, that it can grow at a really, really rapid rate. Same thing is going to happen out over here. Okay, so when we talk about, yes, 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 right, right. Right, last algae, it is last algae, chlorella. Okay, so guys, we can say that, advantages is large quantities of biomass. Biomass is nothing but all the things like, all the materials, the organic materials and all which are present in the body. Okay, so we are talking about those especially. Okay, that is nothing but your biomass. So here, where we can say that, large quantities of biomass. Okay, this can be generated in a very short span of time. Okay, guys, apart from this, the next thing is when we talk about your bacteria. Okay, always remember these microorganisms, they can always be easily modified. Okay, so if you feel that, let's say you own a dairy and in that dairy you are making that Amul Masti Dahi. Okay, for which you are going to add some culture. Let's imagine you don't have this culture. Then what? Okay, then it is not really going to be possible to get the exact same, the structure, the physical qualities, the morphological qualities. It is not going to be possible. Okay, so basically, but then what happens, sometimes you use the microorganisms over and over and over again. So sometimes they undergo modifications or because they have been exposed to like different kinds of your environmental conditions. Okay, so they start undergoing like, rapid mutations as well. So you need to keep a track of all that part. Okay, so I'll just say that these microbes, they are going to be easily modified. So you need to keep an eye on this thing. Okay. Third thing is, you know, when we are going to go for your SCPs, for SCPs, we don't require any specific substrate. That no, this substrate has to be like, this much only. Needs to have this much amount of this protein content, or fat content, or your, sugar content, etc. No, it is not going to be that way, guys. There is multiple varieties of the food sources for your microbes. Okay, fine. And guys, finally, why are we going to eat this thing? Because they are going to be rich, or they are going to show you increased amounts of your vitamins. It is going to be your amino acids, minerals. Okay, so along with this, we are going to have some fibers, etc., etc., etc. So for all these things, guys, we are going to go for your SCP. So this is what we need to know about the chapter, guys. Okay, and with this, we are done with your entire chapter, that is food production and enhancement, food enhancement and food production. Guys, once again, to give you an idea. Oh, just a second, just a second. A broad variety of raw materials. It is third point is broad variety of raw materials. So when we talk about raw materials, guys, it can be, we can grow them on some wood shavings. We can grow them on, for example, like your corn, or for example, we can use them like sugar molasses, like I said. Okay, all these are going to be raw products. Okay, the raw materials. We can grow them on a variety of them. So some people, they will grow this thing on your potato washings as well, that way. Okay. Acha, 1 minute. The font is so small. Okay. Okay. Acha. Okay. Okay. Anyways. Okay. So, guys, this is what we need to know about your entire this thing. That is your food enhancement and production. Okay. So, guys, let's quickly just move ahead and solve a few questions. Okay. 1 minute. Let me just quickly go back to your. I've got taken some questions from your textbook. I just 1 minute, 1 minute. I've got it. 1 minute, 1 minute. Yeah, I don't think so. We have solved questions from your this thing, right? From your textbook. We'll just solve a few, like the MCQs which are there. Okay, the written part. Okay, I mean the long answer questions, like all the thing we have already like put it down. We have it in our notes, so nothing to worry. Okay, but I want you to just go through this once, and let me just see. Okay, now guys, and one thing I want you to remember. Okay, so see, some of you may not be giving neat. Okay, I don't know. Okay, see, even though this is a neat batch, some of you may be like still in a dilemma or something. So guys, whatever we have done, okay, that portion is actually deleted from your 12th standard this thing. Okay, from your exam. From your 12th standard exam point of view, what do you have? You have from this chapter only microbes in human welfare. Okay, otherwise economic importance, food enhancement and your production, that has been removed off, and even your animal husbandry has been removed off. Okay, so just be aware of this thing, guys. Okay, anyways, let's try to quickly just solve a few this thing, and then we'll move on to your mtg, that part. Okay, so one minute, let me have your, in the chat box is open only. Okay, chalo. So guys, let's try to see the first question out over here. Antibiotic chloromycetin, guys, it is going to be obtained through who? Quickly, if anybody remembers chloromycetin. Actually, it is very simple, but then guys, do you remember chloromycetin? Yes, Parnavi has given me the right answer. Parnavi is right. Anybody else? Anybody else? Come on, come on, come on, guys. Anybody? In your antibiotics, we have talked about them. We talked about 4 or 5 antibiotics which are going to be produced. No, 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 no. Only this one is right. Okay, Sia Fanche has given me the right answer. No. Okay, guys, who are we talking about? We are talking about your Streptomyces venezuelae. Okay, so this is the right answer. Streptomyces erythraeus. This is going to give you your erythromycin. Penicillium chrysogenum. This is going to give you penicillin. Streptomyces griseus. It is going to give you streptomycin. Okay, so answer is Streptomyces venezuelae. Okay, removal of large pieces of floating debris, oily substances, etc. during sewage treatment is called, yes, is called what? Yes, very simple, guys. Yes, almost everybody has given the right answer. This is nothing but your primary treatment. Okay, next question is, which one of the following is a free living bacterial biofertilizer? We are talking about bacterial. Okay, so free living. Can anybody tell me quickly? Free living bacterial biofertilizer. No. Yes, Ashita has given me the right answer. Waiting for the others. Himanshu is also right. Sia is also right. Yes. Who are we talking about? We are talking about azotobacter. Guys, what did we say? In your soil, you're also going to have azotobacter, which is going to be a nitrogen fixer. Okay. However, it is not going to show you symbiotic relationship like your rhizobium. That is the only difference. Okay, next one. Okay, most commonly used substrate for industrial production of beer is industrial production of beer. Yes, very nice. Yes, almost everybody is giving me the right answer. Yes, that is nothing but barley. Okay, next one. Ethanol is commercially produced through a particular species of. Yes, guys, what do you think? Ethanol is produced by, yes, it is going to be your Saccharomyces. Okay, one of the free living anaerobic nitrogen fixer. Okay, now guys, this is very important. Okay, we are talking about free living anaerobic nitrogen fixer. Okay, so guys, just think about this thing. Yes, guys, just think. No, 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 no. It has to be free living, guys. It has to be free living. Okay, 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 okay. Okay, I actually, guys, yes, very nice. Some of you have still tried to give me the right answer, but actually, guys, it is a rhodospirillum. Okay, so keep this thing in mind, guys. Okay, it is an aerobic one, rhodospirillum. Okay, microorganisms also help in the production of food, like what? Bread, alcohol, beverages, vegetables, pulses. What do you think? Yes. Okay. So just think, guys. Yes, everyone. Actually, if you look at it, we are going to see that both the options are actually right. It is going to be both A and B. So bread is also right, and alcoholic beverages is also right. Okay, chalo. Next one. Okay, last name. Okay, Moet. Okay, so next one is Moet is a technique used for dash. Yes, guys, Moet is a technique used for what? Very simple, guys. What is the full form of Moet? Multiple ovulation embryo transfer. Yes. Sonali has given me the right answer. Waiting for the others. Guys, this is directly not related. Okay, but then, you know, this is just to give you an idea. What can this be used for? No, 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 no. Guys, no. This is going to be for production of hybrids. Okay, however, this is not the real reason. What have we said? We for more technique, you need to have like super ovulation of the female. Okay, however, anyways, chalo. So this is going to be production of hybrids. Take care. Mule is the outcome of inbreeding, artificial insemination, interspecific hybridization, and outbreeding. What do you think? Someone is saying yes, yes, yes. Almost everybody has given me the right answer except for a couple of them. Okay, yes. What is this? This is nothing but interspecific hybridization. Yes. Okay. Chalo. So, guys, after this, okay, there is one more this thing. We will just do this one. Okay, so we have fungus dash A commercial product is penicillin. Can anybody tell me what is this A? The fungus, the name is dash. Yes, it is your Penicillium notatum. Guys, this is going to be Penicillium. No, sorry, sorry, sorry. This is for commercial product, na? So this is going to be Penicillium chrysogenum, guys. Okay, because of original the found for the foundation or the invention was in your Penicillium notatum, but for your mass production, guys, we have used Penicillium chrysogenum. Okay, bacterium Acidobacter aceti. And what is this, guys? What is the commercial product? Very nice. It is going to be acetic acid. Yes. Okay. Next one, dash Aspergillus niger citric acid. What is this? C. Once again, no, no, no. This is not Saccharomyces cerevisiae. C is fungus. Simple, ekdam simple. Okay, we have to. This is going to be your fungus. Okay, and finally we have yeast. Yeast, what is the name? Yes, Sia had given me the name for this thing. It is nothing but your Saccharomyces cerevisiae. Okay, so this is, oh sorry, this is it, I guess. Okay. Anyways, guys, what we will do over here is right now it is reaching 8 o'clock in my watch. We'll take a quick 5 minute break. And then after that, after coming from the break, we'll start with your that MTG solving session. Okay. Chala. So guys, 5 minutes break. Okay, so there is a question. I had a doubt about CET, that as there is some state word portion deletion, so is it followed by CET? Yes, for CET is going to follow exactly the same thing. There is no change in it. Okay, so whatever portion that has been edited off from your this thing, the books, textbooks, that will not be asked in CET. So don't be worried about that. Oh, sorry, sorry, sorry. Yeah. 1 minute. There is something. Okay, just a second. Okay. Oh, just a second. There was some थोड़ा सा एक सेकंड गड़बड़ हो गया मेरे से. Guys, from here we have solved the animal breed, this thing, right? Animal husbandry, have we solved from here? Just quickly tell me. Animal husbandry. Yes, ना हाँ. Okay, okay. ठीक है, ठीक है, ठीक है, ठीक है. Uh, no, no, no. We haven't done this part from here. The plant breeding techniques, right? So we've just only the animal husbandry one. So that is why I'm like, the first I was just checking that one only, like, like have we solved this thing or no? So I was like, okay, okay. Yeah. Yeah, just a moment, one more picture and we are done. Okay, chalo. So guys, let's try to see the plant part out over here. Okay, the plant breeding techniques one. Okay, so the earlier one we have already done this thing, so not an issue. Okay, chalo. So guys, let's try to see this thing. Okay, so first one, guys, and remember these are only your representative ones. Okay, so we are just selecting the ones which have like come in previous papers, and not only those are the questions which are being discussed. If you have the original MTG, in that there are going to be several more questions. Okay, but those are like dependent upon like what we are doing out over here. Okay, so this is nothing but a representative set. Okay, chalo. So guys, let's try to see this thing and let's see if you can get this thing, get this part. Okay, chalo. So is it still very tiny? You can't see this thing, guys? Can you see this or no? Now, now can you see it? Uh, Devank, is it okay? Take it, fine. Chalo, chalo. Okay, okay, chalo. Okay, so guys, a true breeding plant is okay. Guys, think about this now. Even throughout your Mendels, through your entire Mendel's thing, we have always said pure breeding, pure breeding. Okay, so tell me. Okay, so true breeding plant is what? Yes, Himanshu has given me the right answer. Yes, very nice, very nice. Even Devang has given me the right answer. Yes, Komal is also right. It is going to be near homozygous and produces offsprings of its own kind. Near homozygous meaning it is going to be almost the same as your parent plant. Okay, so that way. Chalo. Okay, next one. Okay, a system of rotating crops with legume or grass pasture to improve soil structure and fertility is called. Now, guys, why do we do this thing? Is see generally in Ha. So generally, what happens, guys? So, like, see, every now and then, if you start growing plants, cultivating plants onto your soil, then the fertility of the soil is going to decrease. Okay, then what happens is like we need to keep on improving the fertility of the soil. So what are the farmers going to do? They are going to bring about this rotation of your crops. So first there may be like a normal crop, then the second plant they are going to grow is a legume, then the third plant is something else, then the fourth plant is a legume, then fifth plant is something else, that way. So that is how they are going to maintain the this thing, the rotation culture. Okay, so basically who is this going to be? Strip farming, shifting agriculture, contour farming, lay farming? Yes, what do you think, guys? What should this be? Shifting agriculture. Okay, 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 okay. Yes. Mm-hmm, mm-hmm, mm-hmm. Anybody? No, 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 no. Mhm. Okay. Now, actually, guys, the answer is your lay farming. Okay, keep this thing in mind. This is a tricky question because most of you, what did you think, guys? Oh, this is shifting agriculture. Guys, no, 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 no. This is not shifting agriculture. Shifting agriculture is something different. Okay, so generally when we talk about your uh, shifting agriculture, okay, what happens is basically you are going to like it is almost on the same lines only. Okay, but then you are going to like preserve the quality of this thing by making use of certain plants only. Okay, so for example, a plot which was like temporarily cultivated, and then maybe it has been abandoned, because of some or the other reason. So that is somewhat you're shifting agriculture. Okay, so basically shifting agriculture is more or less on the same lines, but a little different. Okay, a little different. Okay, so it is this way. A contour farming is basically if you have ever seen on the contours of your hills and all, guys, that is nothing but your contour farming. Okay, also one more thing, guys, in your shifting agriculture. Okay, what people do is in the first case, you will see that they will burn all the crops. Okay, they will burn all the crops, and then after burning all the crops, then they will grow it for a little while. Okay, and then that part is going to be abandoned, then they are going to move to some other place. So that is you're like shifting agriculture once again. And then basically okay, and finally we have one strip farming. Strip farming is basically, guys, when you are going to have the farm which is divided into your long rows or long columns, and you are going to grow plants in your small strips. That is your strip farming, generally done for your low small plants and all. Yes, something of that sort, Himanshu. Yes, absolutely right. Okay, anyways, chalo. Next case, guys. Okay, a cell at telophase stage is observed by a student in a plant brought from the field. He tells his teacher that the cell is not like other cells as telophase stage. At the telophase stage, there is no formation of cell plate, and thus the cell is containing more number of chromosomes as compared to the other dividing cells. This would result in somaclonal variations, polyteny, aneuploidy, polyploidy. Very simple, guys. Very simple. We have done this kind of thing. Yes. Yes. Very nice. Himanshu has given me the right answer. Sneha Singh is right. Waiting for the others. Guys, everybody should participate. Okay. Everyone should participate in this thing. Yes, it is going to give you polyploidy. Guys, can you tell me any one chemical by which we can induce polyploidy? Very nice, Sneha. Okay, it is colchicine. Very good, Sneha Singh. Okay, in plant breeding programs, the entire collection of plant seeds having all the diverse alleles from all the genes in a given crop is called as. So I told you guys, when we did this plant breeding, we had the first step itself, collection of your variety. And it is going to consist of all the different kinds of genes and alleles that are going to be present in the wild type. What is that called, guys? Anyone? Come on. No answer as of yet. Yes, Neha Singh has given me the right answer. Waiting for the others, guys. Come on. Everyone. Yes, Sonali is also right. Guys, what is that called? It is nothing but your germplasm collection. Okay, it is your germplasm. Don't forget this thing, guys. Okay, Green Revolution in India occurred in. Yes, guys. It was no. Only Arya has given me the right answer. It was Green Revolution 1960s. Okay. A collection of plants and seed having diverse this thing. Okay. Yes. What is this? Once again, guys, it is the same thing. Okay. It is your germplasm. Okay. Jaya Ratna developed for green revolution in India are varieties of what? See, I had told you guys. Okay. Jaya, Ratna, Padma, Kanti, etc. Okay. You need to remember this thing. What are the varieties of Guys, yes, very nice, very nice. Almost everyone is giving me the right answer. These are nothing but varieties of rice. Okay, himgiri. Okay, himgiri developed by hybridization selection for disease resistance against a rust pathogen is a variety of. I told you how to remember himgiri. Himgiri is HH. Okay, so this is going to be against Hilbunt. And what is this, guys? This is nothing but, yes, it is going to be a variety of your wheat. Yes. Okay. Next, breeding of crops with high levels of minerals, vitamins, and proteins. What is this? Yes, guys, quickly, quickly, quickly. No, no, breeding of crops with high levels of minerals, vitamins, and proteins is called. Yes, Sia has given me the right answer. Guys, come on. Prachi is also right. Okay, this is nothing but we are going to see this in biofortification only. Okay, next, haploids are more suitable for mutation studies than diploids. This is because, yes, guys, just read this thing. Very simple. Yes, come on, guys, come on, answer, answer quickly. Yes, very nice. Okay, Sia has once again given me the right answer. What is this? Now, guys, always remember all mutations, whether it is dominant or recessive, immediately they are expressed in your haploids. Okay, because haploid is going to have only one set of your chromosomes. So if you receive a dominant allele or if you get a recessive allele, you are going to suffer from it. Okay, so yes, that is going to be your 34. Okay, next one. Which of the following is linked to the discovery of Bordeaux mixture as a popular fungicide? Yes, guys, can you take a wild guess? Okay, no, actually, it is like powdery mildew or downy mildew of your grapes. Answer is actually that, guys. Okay, so keep this thing in mind. Okay. Okay, consider the following four measures A to D that could be taken successfully to grow chickpea in an area where bacterial blight is common. Okay, so guys, just see. Spray with Bordeaux mixture, control the insect vector of the disease pathogen, use of disease-free seeds, use of varieties resistant to diseases. Yes. Which of the two above mentioned, which two of the above measures can control the diseases? Yes. What do you think, guys? Who is it going to be? Yes. Komal has given me the right answer. So, guys, see, what are we going to do? See, we need to see, consider the following four measures A to D that could be taken to successfully grow chickpea in an area where bacterial blight disease is common. Okay, so guys, see, first of all, you can make use of a Bordeaux mixture. This that is fine. Okay, this is right. And then one matter control use of this thing. Use of varieties resistant to the disease. So answer is B, A, and D. Yes. Okay. Next one. In maize, hybrid vigor is exploited by. Very simple, guys. Once again, very simple. How are you going to get hybrid vigor? What is hybrid vigor, guys? When the characteristic of two different parents, they are going to join together, that is called as hybrid vigor. Yes, very nice, guys. Okay, yes, what is it? It is going to be crossing of two inbred parental lines. Okay, chalo. Next one. This is very important. Okay, guys, this question has come up again and again. Okay, in hexaploid wheat, the haploid and the basic number of chromosomes are. Okay, you need to remember this thing. So, guys, when we talk about this thing, okay, when we talk about your wheat, okay, no, okay, wheat, India asking, huh? Okay, so guys, when we talk about wheat, what is the chromosome number of wheat? Okay, so first let's try to understand this thing. Okay, so does anybody have any idea wheat chromosome number? Anyone? It is 42. Okay, and remember it is known as your triticum. Okay, triticum, estivum, or it can be different forms of your triticum. Okay, but now let's try to see. So guys, when we talk about this hexaploid thing, okay, wait. I'll just. Acha, you want to try? Okay, just try it and see, guys. Okay, anybody? Guys, remember this is very important. Very important things. Okay, so let's try to understand what is exactly going to happen. So, guys, you are going to have like this thing. 1 minute. There's a question. Sir, I guess C for now. Okay, fine. Okay, I'll just explain it to you how it is going to be. Okay, so guys, actually, we said this is going to be hexaploid. Okay, so basically, what is going to happen is let's try to understand. Okay, so hexaploid, guys, 2n. Okay, now remember, this thing is going to be 2n, but we are saying that this is going to be hexaploid. Okay, basic number. Okay, so basic number is going to be your 6x. Okay, that is because it is hexaploid, and this is going to be 42. So, guys, when we talk about this X, the basic number of chromosomes, we say X is equal to 42 by 6. X is equal to 7. Okay, this is your this is going to be your basic chromosome number, which we have got from this thing. But now, guys, what is the haploid set? Okay, so let's try to understand what is the haploid set. How is this going to be? So basically, guys, absolutely right. Very nice. Very nice. Can you tell me how did you calculate that? Very nice. Simple. So guys, when we talk about 2n, so I'm just saying 2n is equal to 42, right? So 2n is equal to 42. Therefore, n is equal to 42 by 2. That is n is equal to your 21. So now see guys, what is the chromosome number? Yes. So haploid number, it is n is equal to 21 and x is equal to 7. This is the right answer. Okay, has everybody understood how we have got this thing, guys? Okay, so make sure you know this thing, because these kinds of sums they will also ask. Now guys, what was the most important thing out over here? The most important thing was to know the chromosome number of wheat. If you don't know the chromosome number of wheat, तो ये तो गया ही. तो you just by looking at the options you'll never get what it is. So you need to know the chromosome number. Okay, crop plants grown in monoculture are, okay, monoculture meaning you are going to grow only the same. Acha, okay, how 2n came, didn't get it. 2n, nahi, the second thing that we calculated was n. So once again, okay, chalo, let me just explain it once again. So see guys, when we talk about your hexaploid, okay, so when we say what is this hexaploid, hexaploid meaning 6. So let's say we are talking about 6x because it is hexaploid. What is the chromosome number of wheat? The chromosome number of wheat is 42. So therefore we said, okay, if this is hexaploid, what is the basic, like x, like, hexaploid meaning actually there is, like, 6, right? Okay, so x is equal to 42 by 6. So this is going to be 7. Okay, you got this thing? And the second one, basically, see, second one, what did we say? See, what is this 6x? But 6x is also going to be your diploid. The plant is actually going to be diploid. That is what we also say, right? This 6x is nothing but the plant is hexaploid, but then at the end of the day, it is going to be diploid only. So we say this 2n is also going to be 42. Therefore, n is equal to 42 by 2, and that is equal to 21. So that is your haploid set that way. Okay, chalo. Next one quickly. Crop plants grown in monoculture. Yes. What do you think, guys? Okay, some of them they are saying low in yield. Some of them they are saying characterized by poor this thing, poor root system. But actually, guys, the thing is, remember monoculture. Okay, this is basically against your diversity. Okay, and what you're going to see is that if you have only one type of plants, growing in one particular area, then the chances of pests and on is really really high. Okay, so yes, highly prone to pests. Okay, then in maize hybrid vigor is exploited by. Very nice, very nice. Once again, it's a repeat question actually, guys. Was waiting for the others to answer, but nobody answered. Only three people answered. Guys, come on, you need to participate, guys. Everyone, you need to participate. Okay, otherwise this is not going to work. Okay, triticale. The first man-made cereal crop has been obtained by crossing wheat with. Okay, guys, this is also very important. Once again, yes, very important, guys. Okay, triticale is nothing but your wheat, wheat, guys. So this is going to be, yes, it is. No, 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 no. Guys, keep this thing in mind. Very important. This is going to be done with your rye plant. Okay, and then the combination of two, we got it as this thing. Okay, chalo. Then three crops that contribute maximum to global food production are. Yes. Okay, global food production. Someone is asking me rye plant. Okay, rye plant is basically, once again, just like how we have wheat, no? Just on the same lines, we have your yeah, rye. Okay, so basically it is on the same lines, exactly on the same lines like your wheat. Okay, so guys, coming back. Okay, so which three crops? Okay, so the answer for this, guys, it is going to be A. Those who have messaged me A, very nice, guys. It has contributed extensively for your global food production. Okay, next one. Which of the following is generally used to induce mutagenesis? Yes, guys, which is it? Maize, because maize is like, one of the most important producers, guys. In fact, in Australia, okay, maize is only grown to feed the pigs. Okay, it is huge quantities only used to feed the pigs. Okay, so it is that way. So yes, that is right. Okay, next one, guys. Next one. Okay, so which of these is generally used for inducing mutagenesis in crops? X-rays, UV rays, gamma rays, and alpha particles. Yes. Okay. Now, guys, our general tendency would be to go for your UV radiations. Okay, but remember, no, UV radiation is going to produce undesirable mutants. So, what are you going to do? You are going to go for your gamma rays. Okay, these are more better in generating stable ones. Okay, UV will give you mutants which cannot be used. Gamma rays, see, because see, ultimately these plant products you have to eat them, so you don't want some mutants and all going into your body. So that is why gamma rays. Okay, next one, the name Norman Borlaug is associated with very simple, guys, you don't even have to think. Norman Borlaug, it is, yes, it is Green Revolution. Okay, next one, India's wheat yield revolution in the 1960s was possible primarily due to, yes, guys, kindly quickly answer. No, 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 everybody is wrong as of yet. Yes, anybody? Yes, anybody? Yes. Actually, the answer is this is going to be because of your quantitative trait mutations. Okay. When a diploid female plant is crossed with a tetraploid male, the ploidy of the endosperm in the cell, uh, in the resulting seed is. So, guys, we need to think about this. Okay. So, think about this. Okay, when a diploid female plant, okay, so the female plant is diploid. Okay, so normally, and we have to see what is the ploidy level of the endosperm cells. Okay, chalo theek hai. So this, she is diploid, so that is going to be fine. So you are going to have your, okay, chalo. Okay, so basically inside your ovum, you are going to see a situation like this. You're going to have this, like the embryo sac basically, and inside this at the top, we are going to have your the egg cell, and we have your synergids, and the other side we are going to have your antipodals. Okay, actually we have like anatropous ovule, orthotropous ovule. So depending upon that, the position keeps on changing. But just understand the concept. Okay, okay, so we have the secondary nucleus out over here, right in the center. This is going to give us your this thing. That is, the secondary nucleus is going to give us your endosperm, and this the orange one that I'm showing, that is going to be your egg cell. Now this is a diploid female plant. So guys, this everything inside the embryo sac is going to be haploid. So this is haploid, and this is also haploid. Okay, but now guys, let's imagine tetraploid male. Now, when you have a tetraploid male, at the time of your pollen grain formation, is each pollen grain going to receive tetraploid this thing like tetraploid characters? No. Okay. So what are you going to say? This pollen grain, basically, this is going to undergo meiosis, and by meiosis, sorry, okay, you are going to get this as 2n. So 2n and n, ultimately this is going to be your 3n in the endosperm. Oh, sorry, this is going to be n 2n, sorry, 2n. Okay, this is 2n, and rest everything is n. So 2n, sorry, 2n plus 2n, it is going to be 4n. Actually it is tetraploid. Okay, sorry, it is tetraploid. Okay, so guys, keep this thing in mind. Very, very important question. Okay, very, very important. Okay, and the basis of this thing, guys, this is I your teacher must have done this thing, like, the structure of your the ovule, how it is going to be. So it is based on that. Okay, so everything is going to be your N except for your secondary nucleus, which is going to be 2N. Okay, so guys, just keep this thing in mind. Sure. Okay, if a diploid cell is treated with colchicine, then it becomes, yes, what is it going to become quickly? Yes, it is going to become tetraploid. Which of the following crops have been brought to India from the New World? Yes. Now, guys, this is going to be a little this thing, like a general knowledge kind of a question. So, yes, it is going to be yes. Very nice, very nice. It is going to be cashew, potato, and rubber. Okay, but you have to keep this thing in mind, guys. Okay, next question. Before the European invader, which vegetable was absent in India? Very nice question, guys. Very nice question. Can anybody guess this thing? Very nice. Devank has given me the right answer. Absolutely right answer. Answer is potato. Guys, potato and tomato. Okay, they were not present. Only after the Europeans came here, that time we got this thing. Okay, triticale is obtained by crossing wheat with, once again, it is your right. Repeat question. The reason for vegetatively reproducing crop plants to suit for maintaining hybrid vigor is that, yes, guys, what do you think? Is that they are very nice. Okay, Komal has given me the right answer. Okay, that once the desired hybrid product, I mean, you have got it, there are no chances of losing it as long as you maintain that thing properly. So that is why, yes, again, the second answer is the right one. Once the desired hybrid is produced, no chances of losing it. Which of the following is the new world spice that has become an essential part of Indian cuisine? Red pepper, black pepper, and cardamom. Yes. What do you think, guys? Once again, you will have to take a little guess out over here. Okay. But actually, it is red pepper. Yes. Oh, sorry, I missed this 53rd one. Sorry, sorry. Okay. Yes. Um, guys, if an angiospermic male plant is diploid and female plant is tetraploid, the ploidy level of endosperm, once again, it is going to be what? Okay, yes. So can anybody think about this? Yes, yes. It is exactly on the same lines like we did, guys. So that is going to be pentaploid. Very nice. Very nice. Okay, haploid plants can be obtained by culturing. Now, very important, guys. This had recently come in one of the papers. Okay, so keep this thing in mind. Haploid plants can be obtained by culturing what? Pollen grains, root tips, young leaves, endosperm. What do you think? No, you have to. Yes, Neha Singh has given me the right answer. Devank is also right. Guys, you have to see who is haploid from amongst these. Who is haploid? It is going to be your pollen grains. So answer is pollen grains. Okay, chalo. Next one, 56. Okay, haploid plants are preferred over diploids for mutation study. Okay, because yes. Okay, once again, just see what is the answer. What do you think, guys? Yes, very nice, very nice. Some of you are giving me the right answer because recessive mutation is expressed immediately. You don't have to wait. Okay, it is going to be very quick. Okay, anyways, which crop variety is not due to induced mutations? Yes, very nice, very nice, very nice, very nice. Okay, yes, Prabhat of Arhar. Okay, guys, very important. Okay, just keep this thing in mind once again. Very nice, guys. One minute, 1 minute. Which property is not due to? Oh, sorry. No, not due to. Not due to. These are due to induced mutations. Okay, like we have Aruna of Castor. Sorry, guys. Those who have given me D is right actually. Aruna of Castor is the right answer, guys. Okay, next one in crop movement program, haploids are important because yes. Once again, this is you have to think. Yes, once again, some of you have given me the right answer. They perform, I mean, they form perfect homozygous. That is right. Okay, but anyway, this is a very old question actually. Okay, not recently seen anyway. Okay, the next one, an algae that is alga, which can be employed as food for humans. Human beings is Eulothrix, Chlorella, Spirogyra, and Polysiphonia. What do you think? Who is it going to be? It is going to be, yes, Chlorella. Okay, now even your Spirogyra can be used. Okay, but Chlorella is a better version of it. Okay, then consider the following four statements and select the option which includes all the correct ones only. All correct ones. Okay, single cell spirulina can produce large quantities of vitamins, minerals, proteins, etc. Body weight wise, the microorganism Methylophilus, Methylotrophus may be able to produce several times more proteins than cows per day. Common button mushroom are very rich source of vitamin C. A rice variety has been developed which is a very rich in calcium. Yes, guys, what do you think? Okay, which includes all the correct ones. Okay, so the first one is actually right. The second one is also right. Okay, so answer should be D for this thing, guys. Okay, answer should be D for this. Okay, next. Okay, this is very simple, guys. Yes, absolutely. Very nice, Himanshu. This is used by astronauts. Absolutely right. Okay, a protoplast is a cell. Guys, quickly, like very simple. No. Someone has just answered without looking at the question properly. A protoplast is a cell which is without what? I just told you. Guys, we just revised that part as well. It is, Hurry, come on. It is without your cell wall, guys. Some of you are saying without nucleus. How is it going to be without nucleus? If it is without nucleus, guys, it is going to cease to exist as a cell. Okay, chalo. Next, a technique of micropropagation is okay. So protoplast fusion, embryo rescue, somatic hybridization, somatic embryogenesis. Yes, what do you think, guys? Actually, the answer for this thing I will only give you. It is somatic embryogenesis, guys. Okay, anyways, which of the following enhances or induces fusion at the time of protoplast? Yes, guys, quickly think about this. Yes, anybody wishes to try. Guys, come on. You have to try, guys. This is actually PEG. We had talked about PEG, guys. Okay, this is going to take place in the presence of PEG, that is polyethylene glycol and sodium nitrate. Okay, guys, keep this thing. Okay, keep this thing in mind though. Okay, to obtain virus-free healthy plants from a diseased plant, one tissue culture technique. Okay, which part is going to be taken? Yes, I told you guys. Which of the part you're going to take, especially you are going to take? Yes, especially it. I told you it has to be your meristem. But now see, look at the options out over here. Apical meristem. Yes, here they are also talking about axillary meristems. So yes, whatever meristem that you're going to get, that is going to be used. Okay, so you are going to have apical and axillary meristems. Okay, the next one is Tissue culture technique can produce infinite number of new plants from the. Yes, okay. Economic importance of this technique is in the raising. Okay, tissue culture technique can produce infinite number of plants. Okay, so let's try to see the options, guys. Yes, genetically uniform population identical to the original plant, homozygous diploid plants, new species, variants through picking up somaclonal variations. Yes, what do you think, guys? Yes, actually, sorry. Yes. Okay. Uh, चलो ठीक है थोड़ा सा ही है we can just finish this thing off. Okay, really quickly. Okay, which of the following statements is not true about somatic embryogenesis? The pattern of development of somatic embryo is comparable to that of zygote. Okay, somatic embryos can develop from microspores. Somatic embryo is induced by using auxins. A somatic embryo develops from a somatic cell. Guys, what do you think? Okay, so I will only give you the right answer. Somatic embryos can develop from microspores. Okay, as we move ahead, guys, we are going to see something, okay, called as your apospory, apogamy, okay, and all that stuff. Okay, which part would be most suitable for raising virus-free plants from micropropagation? Okay, bark, meristem, vascular tissue, and none of the above. Yes, what do you think, guys? Yes, it is going to be your meristem. Very nice. Soma clones are obtained by very simple. You don't even have to think about it. It is by your tissue culture technique. Okay, in order to obtain virus-free plants through culture media, the best method is, yes, guys, think about this. What is the best method? Yes, it is meristem culture. Okay, the technique of obtaining large number of plantlets by tissue culture method is called as, very simple, guys, very simple. It is micro propagation. Okay, next one, cellular totipotency is demonstrated by only gymnosperm cells, all plant cells, all eukaryotic cells, only bacterial cells. Guys, what do you think? Yes, very nice. Himanshu has given me the right answer. Anybody else wishes to think about this? Yes, it is going to be all plant cells, guys. Actually, it is going to be all plant cells. Okay, next thing is in tissue culture medium, the embryoids formed from pollen grains are due to cellular totipotency, organogenesis, double fertilization, test tube culture. Yes, what do you think, guys? It is because of cellular totipotency. Then, yes, guys, right. Okay, coconut milk is used in tissue culture in which in which present. Okay, what is going to be present now, guys? Keep this thing in mind. This is the richest source of that hormone. Okay, who is it going to be, guys? Can anybody tell me? Guess. It is cytokinins. Okay, cytokinins. Yes. Cellular totipotency was demonstrated by Theodore Schwann, Leeuwenhoek, Stewart, and Robert Hooke. Guys, if you have to take a wild guess, who is it going to be? It is going to be Stewart, because the rest three we know for them, for sure. And finally, the last one, guys, which ones produce androgenic haploids in anther cultures? Yes, who do you think will they be? Haploid, haploid anther cultures. Yes, it is going to be your young pollen grains. Okay, so guys, see this entire set, whatever we did in the last part and all, it was a little on the trickier side. Okay, because some of them you were not seeing them, some of them we have not come across. Okay, but some of them were like, of your journal types, or the general types, also general knowledge types. Okay, these kinds of questions are going to pop up in your exams from somewhere or the other. Okay, so guys, always remember, do not expect that 1 minute, 66, 1 minute, huh? Which of the following statements is not true about somatic embryogenesis? The pattern of development of somatic embryo is comparable to that of a zygotic embryo. Yes, this is true. Somatic oh no no somatic embryos can develop from microspores. No, 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 no. That is going to be your anther culture. Somatic embryos, no. So for somatic embryos, you have to take two cells and add them together that way. Somatic embryo is induced usually by an auxin. Yes, this is true. And somatic embryo develops from a somatic cell. This is also true. Yes, this is right. Okay, the right matlab, this is the wrong answer. Rest, the other three are true. This is wrong. Okay, so guys, once again, we'll stop over here, okay, with this part, and next class we'll start off with a new chapter. Okay, so bye bye guys, everyone, take care. Bye everybody. Bye bye.